So far, we've seen mechanisms to introduce simple building blocks like cyanide and phosphate from the groups of Catling and Templeton. We've also seen rudimentary ways in which simple chiral molecules, amino acids, and small peptides can spontaneously break symmetry and possibly lead to homochirality from Donna Blackman's group. And we even saw a potential mechanism for the early evolution of translation from research in the supplement group. And so I think it's interesting but conspicuous that we did not yet feature anything about the origin of metabolism or the early evolution of metabolism. And so with today's session, I'm very happy to say that we will touch on that topic, not just touch on it, but go into it in some detail. So today, we're going to have Joris Zimmerman and Quentin Dherbassy from the group of Joseph Moran at the University of Strasbourg. So Joris is currently a postdoc. He started in Joseph's group in 2020 with an MRT fellowship and completed his PhD in 2024. During his PhD, he studied key biological transformations found in core metabolism, especially those in gluconeogenesis, which is how life makes sugars. Now, Quentin, he studied catalysis during his PhD in the group of Francois Colbert and Joanna Wenzel-Delorde in Strasbourg, focusing on the control of axial chirality by CH activation. He then joined the Proctor group as a postdoctoral researcher in 2018, focusing again on asymmetry, this time through copper-catalyzed reactions, borylation specifically. And he joined Joseph's group in 2020, where he's currently studying the prebiotic origins and catalytic functions of biological cofactors, I believe specifically pyridoxal. So what we're going to do is we're going to have Professor Moran give a brief topical introduction. And then Joris will present and Quentin, I believe, in that order. Again, just a reminder, we're going to hold all questions for both speakers until the end for a combined Q&A session. In the Q&A session, excuse me, you can either raise your hand in the Zoom interface and we will call on your name and you can speak your question aloud, or you can put your question in the chat and we'll do our best to address the questions in the order that they appear, and hopefully we'll get to all of them. So without further delay, Professor Moran, take it away.
Great. Thanks, David. I hope I'm just share my screen here. Okay. You see that okay?
Yes.
Great. Great. Well, thanks, Dave, for the invitation to this NASA PCE3 seminar series. I thought I would just begin by giving a little, philosophy that we kind of use to guide our research in the group. And one of those sayings is that life is a process, not a thing. Life is many processes, and some of those processes make things and break them down. But I think there's been a lot of focus in the origin of life field on synthesizing things independently from those processes. And I'm not sure that that's something that actually can be separated. So what we've tried to do is focus on recreating the process and creating the things as a byproduct of that, rather than just making molecules as a separate goal. So to introduce this idea, I'll talk about a process we all know well, which is a fountain. And what is a fountain? Well, there's a pump somewhere off the screen that is burning fuel, and that's allowing water to be driven through a pipe up into the sky. And the reason the fountain and the pool doesn't go dry and all the water go into space is because there's an opposing force acting on that water, which is the force of gravity, of course, and that's bringing that water back down into the pool. And so what makes a fountain really is these two superimposed processes that look roughly like the reverse of each other, but they are not the microscopic reverse of each other. They are driven by different thermodynamic driving forces. And that's what allows the fountain to escape equilibrium and to have its dynamic form that we know so well. So the same is true for life. it's starting from small geochemicals like CO2 and ammonia and phosphate. It's converting them into molecules like carboxylic acids, specifically the keto acids. And with the entry of phosphate into that metabolism, you get sugar phosphates. With the entry of ammonia, you get the amino acids. And a lot of those processes are driven by phosphorylation chemistry. And then the sugar phosphates and amino acids can react together and with C1 molecules to form the nucleotides and coenzymes. And driven again by phosphorylation, those nucleotides and those amino acids can be polymerized, the nucleotides into the genetic polymers, of course, RNA and DNA, and the amino acids into proteins. Now, in the synthetic direction, going up the pyramid, also known as the anabolic direction, when there are redox reactions, they are typically reductive, although not always. And the phosphorylation processes that are usually what drive things uphill, and they are enabled by the collapse of proton gradients across membranes. Now, in the opposite direction, the breakdown direction or catabolic direction, when there are redox reactions, they are typically driven by oxidants, and breakdown is also aided by hydrolysis with water. So like the fountain, there are two opposed processes that look roughly like the opposite of each other, but they're not the microscopic reverses of each other. They are driven by different thermodynamic driving forces. And this is what allows metabolism to escape equilibrium and have this dynamic pattern in nature that has been persisted across deep geological time, as far as we know, back to the origins of life. So when thinking about the origins of this process, it can be helpful, I think, to ask ourselves, how does any self-organized dynamic process emerge spontaneously in nature? So I'll take the example of a hurricane. A hurricane is just made up of air and water, but you only get a hurricane when you apply an energetic stress, which is a temperature gradient. But there are also constraints on that. Not any temperature gradient will do. It has to be the right magnitude, not too big, not too small. It has to occur on the right distance scale. In this case, the 1,000-kilometer distance scale. A centimeter or a light year won't do. And so if we now apply that thinking to self-organized chemistry, yes, there are building blocks that may be important. Geochemicals at the bottom of that pyramid. An energetic stress would be important. Based on how that redox gradient works, one could think it might be a redox gradient. It could be a chemical one, an electrochemical one, or a photoredox one. However, in the last case, it appears to have taken life around 2 billion years to evolve photosynthesis. So it seems a bit strange maybe to propose that photoredox chemistry might have also been what drove that system into existence in the first place. But then there's also constraints. We know that chemistry is greatly influenced by catalysts. We know that prokaryotic cells exist on the micron scale, so scale may also be important. Conditions like pH and temperature may be important. Our lab is starting to think that electric fields might also be important. And there may be other constraints that we just haven't been smart enough to think of yet, but that may be important in the future. So with that in mind, we can think of the origin of life as a bit of a search problem based on building blocks and energetic stresses and constraints. And so we have to decide what is the dynamic pattern that we're looking to actually recreate. And so I would suggest, based on the writings of these people shown here and others, but these are the ones that inspired me, that is actually this basic core of metabolism that may be a remnant of this original self-organized chemical network. There's a couple of assumptions built into that idea. First assumption is that a precursor to this sort of network may have actually existed. I've drawn it out here in a map-like form. That is an experimentally testable hypothesis. And so our group has spent, over the last 7-8 years, we've done a lot of work on different pathways that make up that core of metabolism. And we found that metals, either in the reduced form, in the zero-valent form, or in as metal ions, can actually promote a lot of that chemistry. Not all of it. We've have not got all of it working yet, but a lot of it has been. And I'm just showing mostly the work done by our group, but other groups have also contributed to this area and actually achieved some of the things that we have not achieved. So I think it's a promising hypothesis to continue to explore. Now, the second assumption about this is that there can be some sort of self-complexification of such a dynamic network. through catalytic feedback. So in other words, the products produced by such self-organized chemistry could feedback as catalysts to reinforce reactions that were already happening, in which case that would divert flux within the network and potentially prune the network. But at the same time, those molecules might have new properties that the precursors didn't have because they're more complex, so they have some particular properties that make them more reactive. They could do new chemistry that couldn't have been done before, which could expand the network, or they could do catalysis that couldn't have been done before, and that could also potentially expand the network. So there's these two mechanisms of pruning and expansion that may have been at play simultaneously and provided a primitive form of change over time to this chemistry, even before we had sophisticated mechanisms of evolution like translation and self-replication. So to address that point, our group has looked at some molecules that are produced by metabolism, and especially the coenzymes. And we've looked to see under what conditions they actually can do the jobs that they do in metabolism and promote some of the reactions that may have actually been required for their own synthesis. And what's interesting out of this is that, and Quentin will tell you about this in his talk, what's interesting about that is that they all seem to also depend on metals when they work non-enzymatically. So that's been an interesting observation. So we hear two talks today from our lab. The first one is from Joris Zimmerman, and he'll talk about an alternative strategy and searching for conditions that might promote metabolism rather than searching within one pathway like we had been doing previously. He's trying to search for reactions that appear in multiple pathways, and therefore he can find conditions that might allow more of the network to emerge in a more broader sense. And Quentin will tell you about his work on the non-enzymatic catalysis by PLP. So with that, I will turn over. I'll just thank all the people who contributed to work in our lab, all the funding agencies that contributed, and I'll turn the floor over to Joris for the first talk.
Thank you. I hope everyone can see my screen now.
Yes, we can see it. I
Okay.
like your mural back there. I was one of the people who helped make that mural.
Oh, great. Okay, so good afternoon, everyone. Thank you for the kind introduction and thank you for the invitation. Today, I will be talking about alkene hydration, which is one of the main transformations found in metabolism. But before showing you some results about this transformation, let me first introduce you some key aspects of what we call proto-metabolism. Sorry. So in our lab, we are trying to explain the transition from a non-living world, mainly composed of simple geochemicals such as gases, minerals, and water, to the biochemistry of the living organism. So basically, how to explain the emergence of this complex and self-organized system called metabolism. And thinking about it, you could first ask yourself how to approach and reconstruct this complex system. Well, one approach called the metabolism first hypothesis proposes that an early metabolism called proto-metabolism emerged from chemical self-organization driven by an input of external energy from the primitive Earth. So in this approach, the complex biological catalyst called enzyme would have been predated by some naturally abundant metal or mineral to catalyze the chemical reaction. So the system will have evolved over time with a minimal reconstruction or modification of the initial core structure. Eventually, the system will have formed its own catalyst to decrease the dependency with the environment and to get complexification of the system. So complexification will have occurred by two main mechanisms. The first one would be by reinforcing some existing pathway via autocatalysis. So this will help to prune the network. And the second one would be by turning on new reactions. So this will help to expand the network. So now that we have a better idea of what could be the process of the complexification of this protometabolism, you could now ask yourself how to get the emergence of this protometabolism. Well, here I will try to address that question and illustrate that question using this famous example of the Conway's game of life. So in this programming game, you have a small set of programming rules which are repeated over and over. And here, these rules are dictated by the programmer. And of course, most of the combination of these rules will lead to nothing. But interestingly, some do. And as you can see over time, the repetition of this small set of particular rules produces a complex behavior. So you have more and more complex structures which appear, as well as self-organization. So now if you keep that idea in mind, we can try to connect it to our idea of protometabolism. So by analogy, our protometabolism will have a small set of chemical reaction mechanisms. And here are shown the different mechanisms in question. And this mechanism will have been probably dictated by some catalysts, so things like naturally abundant metal or mineral. And it is the repetition of this small set of particular mechanisms which will have triggered the spontaneous emergence of our protometabolism. So here is a proposed map of this protometabolism that Joseph just introduced. So far, our strategy was to identify the non-enzymatic condition that enable single reactions or a sequence of reactions within the different biological pathways represented here. However, even if this strategy proved to be very successful, it raises the question of compatibility between all of these different reported conditions. So since the metabolism is built up on a small set of repeating chemical mechanisms, we thought that investigating by mechanism type rather than by pathway could help us to identify a unified set of conditions for the emergence of this protometabolism map. So now let's take the example of the hydration-dehydration mechanism. So alkene hydration and the reversible alcohol dehydration is a transformation which occurs 3 times in the RTCR cycle, where fumarate is converted to malate, and aconitate is converted to citrate and isocitrate. But this transformation also occurs 1 time in gluconeogenesis, where phosphoenol pyruvate needs to be hydrated to 2-phosphoglycerate to have the sugar phosphate backbone, which is then incorporated in ribonucleotide. So from here, we thought that alkene hydration might be a good starting point to evaluate our complementary strategy. So here, what you can see is that these 4 substrates have in common that they feature a conjugated alkene with a carboxylate group. So we might expect that they could act as a Michael acceptor for the hydration reaction. And indeed, when you are looking at the biological pathway of these different transformations, you have the carboxylate group, which is activated either by magnesium or by iron sulfur cluster, so that you have the Michael addition to form this intermediate, which is then protonated to form the hydrated product. So now when you are looking at the non-enzymatic variant, which were reported for the 3 transformations found in the RTCR cycle, what you can find is that these transformations were reported in different conditions, sometimes requiring the presence of metal ions, such as zinc or chromium, or sometimes not. So last year, these 3 transformations were reported under a set of common conditions using aqueous microdroplets, which were generated in a mass spectrometer. But in this case, it is not clear whether this study is periodically relevant. So there is still 1 transformation which has non-enzymatic variants, which is the conversion of phosphoenol pyruvate called PEP to 2-phosphoglycerate called 2-PGA. Why is that? Maybe because the non-enzymatic conditions reported so far, which are harsh and acidic, are not compatible with the PEP system. And indeed, when we are looking at this study about the non-enzymatic hydrolysis of PEP to pyruvate, what you can see is that this transformation is favored at elevated temperature under acidic condition or in the presence of metal ion. Exactly the condition which are reported for all of the different hydration. So in this work, we are wondering if we could identify a set of conditions which will favor the hydration reaction forming 2-PGA compared to the hydrolysis reaction forming pyruvate. And we were even more interested to figure out if we could identify a set of conditions which will be compatible for all of the different hydration that you can find in the core metabolism, including for the first time, the hydration of PEP. So here is the system that we want to study. So to minimize the hydrolysis side reaction, we buffer the reaction using sodium bicarbonate to have a pH around 8-9. And the first thing that we did was to screen a bunch of different metal. And when we did that, we found that when we were starting the reaction from iron 0 or iron 2, we were able to detect trace amount of 2-PGA by NMR spectroscopy. So next, we screen a bunch of different additives trying to improve the yield of the formation of 2-PGA. So here I'm showing you a few of these examples. And what you can see basically is that only when we are using polyphosphate as additives, we are able to increase the yield of 2-PGA with, for example, 16% yield when we're starting the reaction from pyrophosphate. So we did the same starting from iron 2. In that case, we observed the same trend, except that we have only half of the yield obtained in that case. So next, we show that we could decrease the temperature from 75 degrees to 25 degrees. And just by increasing the reaction time, we could also get 11% yield of 2-PGA. So now that we identify a set of conditions to study that transformation, we're next wondering what are the key parameters of this transformation. So by studying that transformation, we identified the formation of three main products. So the hydrated product, 2-PGA, the hydrolyzed product, pyruvates, and we also identified the formation of this side product, glycolate, which is another metabolite. So the first thing that we did here was to evaluate the importance of each parameter of the reaction on the formation of this three main product. So we first started by varying the concentration of iron in the system. So when you start from 1 equivalent and you increase to 2 equivalent or 5 equivalent, what you can see is that the yield of PEP is increasing up to 25% yield, while the other side products are not so much modified. You observe the same thing when you decrease the amount of iron to, for example, 0.5 equivalent. In that case, you have only 10% yield of 2-PGA. So from this experiment, it seems that the formation of 2-PGA is linked to the amount of iron which is used in the system. So next, we varied the concentration of sodium bicarb in the system. So when you start from 5 equivalent and you increase to 10 equivalent, you don't have so much significant changes on the ratio of the product. But when you start to decrease the amount of sodium bicarbonate to 2 equivalent, or when you remove bicarbonate from the system, in that case, the formation of the side product is favored. Finally, we varied the concentration of pyrophosphate in the system. When you start from 2 equivalent and you increase to 5 equivalent or you decrease to 1 equivalent, in that case, you don't have so much significant changes. But when you are removing pyrophosphate from the system, in that case, you obtain only trace amount of 2-PGA. So what was interesting here in that case was that when you are removing pyrophosphate from the system, in that case, we observed some precipitates at the end of the reaction. So the same thing was observed when we were using any other additives except the polyphosphates. So from these observations and from the literature, we propose that pyrophosphate and polyphosphate are acting as a chelating agent to the iron species which are generated in our system, so that they will prevent their precipitation and the reaction in that case can occur. So now that we have a better idea of the influence of the different parameters on the system, we're next wondering what is the promoter of this reaction. So when you start the reaction from iron 0, you get 16% yield. Iron 0 in our condition can oxidize to iron 2 and iron 3 species. However, when you start the reaction from iron 2, you get only 8% yield, while when you start the reaction from iron 3, you don't have any reaction. So what was interesting here is that it was reported that when you put iron 0 in a sodium bicarbonate solution, exactly as is it in our condition, what you can see is that iron 0 can form what is called green rust. So it's an iron oxide minerals made of iron 2, iron 3 precipitate, which has a specific double layered shape. So to test whether green rust might be the promoter of our reaction, we made some green rust separately, and then we use it in our system. And when we did that, we obtained 10% yield of the product. So from this experiment, it seems that whatever the oxidation state that at which we are starting with iron, it seems that the best yield is always obtained when we start from iron 0. So one possible explanation might be that iron 0 is also acting in our system as a reducing agent, so that it will help to regenerate the promoter of the reaction and so that the reaction can continue to occur. So to test whether if that hypothesis is possible, we added a reducing agent, zinc 0, in two different conditions. First, in the presence of the iron 3, which was inactive. And in that case, we are able to form now some product. And we also added zinc 0 in the presence of iron 2. And in that case, we were able to increase the yield of the formation of 2-PGA. So from these experiments, it seems that iron 0 could also act as a reducing agent in our system to regenerate probably the promoter of the reaction. And that's why we are observing a higher yield in that case. Now, from this experiment, it seems also that iron 2 or a mix of iron 2, iron 3 species could be the promoter of the reaction. Since so far we are performing our reaction under air, so in the presence of oxygen, we were next wondering how important is oxygen for the system. So to evaluate that, we performed 4 different experiments, starting at different oxidation state of iron. And we performed this reaction in the absence of oxygen. So in that case, what you can see is that we didn't obtain any product. Either when we were trying a different oxidant than oxygen, such as nitrate, we didn't obtain any product. So it seems that oxygen is important for the reaction, and that the reaction will require a combination of iron species and oxygen to occur. In that case, oxygen could be important for the formation of the promoter, or could maybe involve in the mechanism of the formation of 2-PGA itself. So after investigating the promoter of the reaction, we next wanted to investigate the formation of the main side product, glycolate. So what we did first was to follow the reaction over time. And what you can see when you are doing that is that over the first hour, glycolate is formed. And after an hour of incubation, 2-PGA start to be formed while glycolate is not formed anymore. So from these experiments, it seems that these two products are formed via 2 different pathways. Moreover, it was reported that glycolate was formed from a prebiotic alternative synthesis of PEP under oxidative conditions. And in that case, 2-PGA was not reported. So next, we wanted to investigate the mechanism of the formation of glycolate from PEP. So to do that, we use H2O2 to mimic oxidative conditions. And when we started the reaction from PEP in our standard condition, and where we are using H2O2 instead of iron, in that case, we are able to form glycolate and the following side product. So from these observations and from the literature, we propose that glycolate could be formed via the epoxidation of the terminal alkene of PEP using H2O2. Then the epoxide could be hydrolyzed, and after dephosphorylation, you could form hydroxypyruvates, which under this condition could undergoes oxidative decarboxylation to form glycolate. To verify this hypothesis, we started the reaction from hydroxypyruvate. and we put it in the presence of H2O2 in our standard condition, or we put it in the presence of iron in our standard condition. And in that case, after 2 hours of reaction, we observed that hydroxypyruvate was completely converted to glycolate and other side products. So from these experiments, we propose that glycolate might be formed due to the presence of H2O2 in our system, and H2O2 could be formed in situ in our system from the oxidation of iron 0 in the presence of oxygen to the iron 2 and iron 3 species. So back to our PEP system and some last experiment. So what is interesting here is that when you put PEP under these specific conditions, you don't have only the formation of these three main products, but you also have some additional transformation, which is forming the following reaction network. So pyruvate can decarboxylate to acetate, glycolate can further oxidize to glyoxylate. Then altogether, pyruvate and glyoxylate can undergo a sequence of reaction forming alpha ketoglutarate. This sequence of reaction was already reported previously. Glyoxylate will further oxidize to oxalate and decompose to formate and CO2. So we have all of these different transformations which are occurring simultaneously. And even if PEP is decomposed to pyruvate and glycolate, they can be then recycled in the system to form alpha ketoglutarate, which is a key metabolite found in the RTCR cycle. So now that we have a better idea of the system of PEP under these conditions, we are next wondering if this condition could be compatible with the hydration of all of the other core metabolites. So here I'm going to show you three key examples, which also help to define some key parameters of the transformation. So we started with fumarate, which is one of the key metabolites found in the RTCR cycle. In that case, fumarate features an internal alkene compared to PEP, which was featuring a terminal alkene. So fumarate was converted to malate in up to 20% yield, which means that the reaction is not limited to the terminal position of the alkene. So next, we wanted to verify if the fact that we have a conjugated alkene is required for the transformation to occur. So in that case, we increased the distance between the carboxylate group and the alkene function. And when we did that, we didn't observe the formation of the hydrolytic product. So the reaction seems to be selective for conjugated alkenes. Finally, we performed the reaction on aconitate, which is the second substrate of interest. In that case, aconitate features two potential sites for hydration, this site with the carboxylate conjugated here, or this site with the carboxylate conjugated here. And in that case, we observed 90% yield of isocitrate, while citrate was only formed in 3% yield. So from this substrate, it seems that the reaction is favored on the less substitute site of the alkene. And what was interesting when we were studying the system is that we identified the formation of two side products, pyruvate and acetate. So when we saw that, we were wondering if these two products could be the result of the retroaldol reaction of citrate. The retroaldol reaction of citrate, which is another key transformation found in the RTCR cycle and which was never reported so far in bulk water. So here is the system that we want to study. Aconitate can be converted either to citrate or isocitrate. And from a citrate, you could have the retroaldol reaction forming acetate and oxaloacetate, which under this condition could decarboxylate from pyruvate. So to verify that hypothesis, we started the reaction from citrate under our standard conditions. And in that case, we observed the formation of acetate and pyruvate. So to further verify that we have a retroaldol reaction here, we wanted to trap the intermediate oxaloacetate using, for example, a reducing agent. So to do that, we added zinc 0 in our condition. And in that case, we observed the formation of malate. While when we were starting the reaction from isocitrate, we didn't observe any of these products. So from these experiments, it seems that another key mechanism is found to be compatible under the same set of conditions than the hydration reaction. And this is an important finding because so far, the only non-enzymatic variant which was reported of the retroaldol reaction was in these acidic aqueous microdroplets. So as a final experiment in this project, we wanted to evaluate if all of the different hydration that we are performing separately could also occur simultaneously under a one-pot condition. So similar to what you would expect to have for the emergence of a chemical reaction network. So what we did is that we took our three substrates of interest, aconitate, fumarate, and PEP. And when you put them all together in our condition, the first thing that you can see is the formation of the four different hydrated products. Under the same condition, you can have the retroaldol reaction of citrates, which form acetate and oxaloacetate, which further decompose to pyruvate. And from PEP, you can have its decomposition to glycolate, which can be then oxidized to glyoxylate and react with pyruvate via an aldol reaction, forming malloy formate, which can dehydrate to form fumalloy formate, which can then be reduced to form alpha-ketoglutarate. So from this key experiment, we found that all of the different hydration reactions were compatible under a set of conditions in a one-pot manner. So you don't have a selectivity for one of the substrates. All of them can occur simultaneously, and the formation of the hydrated product were roughly in the same concentration, except for citrate, obviously. And finally, we're able to develop a continuous reaction network, which contain different types of chemistry, such as hydration, dehydration, aldol and retroaldol reaction, and reduction and oxidation, all of these transformations occurring simultaneously. So now to recap with this key transformation about the alkene hydration, we first found that the reaction was requiring a combination of reduced iron species and the presence of pyrophosphate. But this transformation also requires the presence of oxygen for the reaction to occur. We found that the reaction was selective for conjugated alkenes, just like the biological mechanism. And we also found that when you have two potential sites for dehydration, the hydration is favored on the less substitute site of the alkene. More importantly, we found a common set of conditions which was compatible with all of the different hydration found in the core metabolism, including for the first time PEP, and which was also compatible with another key transformation of the Artesia cycle, the retroaldol reaction of citrates. So now let's take a few minutes to discuss about the prebiotic relevance of this study. So we have two key transformations which can occur under the same set of conditions, which require a temperature range of 25 to 75 degrees and basic conditions. It also requires the combination of iron species, polyphosphate, and oxygen. So iron is one of the most abundant metals on Earth and is known to play a fundamental role in biology and was shown to promote multiple non-enzymatic metabolic reactions. Moreover, carbonate green rust, which could be formed under our set of conditions, is a natural abundant mineral which may have covered the primitive ocean, the floor of the primitive ocean. Next, polyphosphates, they are known to be present naturally in some mineral, and they are known to be produced by geological processes such as volcanoes. So these three parameters seem to point toward the same place, which could be alkaline hydrothermal vents. These vents are known to be anaerobic and reducing environments. So what about oxygen? Well, recently, and more and more studies are showing now that oxygen could be formed under anaerobic conditions and in a deep ocean via a different mechanism. So it could be formed via water hydrolysis or via friction from the water flow against the surface of the minerals such as iron or silicate-based minerals. Or a last mechanism could be the electrochemical water splitting over some metals. So oxygen could be very locally and continuously formed under anaerobic conditions. So the study of these two key transformations seems to point toward alkaline hydrothermal vent as a potential environment for the emergence of our photometabolism. So the next step in this work will be to investigate these different transformations and trying to find some non-enzymatic conditions and see if they point toward the same environment as the other one found. So with that, I would like to thank Joseph for his guidance and his supervision during my PhD, and I would like to thank my colleague and my former colleague for their help during my PhD. And I would like to thank you for your attention. With that, I think, Quentin, you can now.
Thank you very much, Joris. Quentin, whenever you're ready.
Yeah, so are you seeing the right screen?
We're seeing it in presenter mode.
Okay. Oh, okay. Well, I went wrong. Should be good.
Looks good.
Excellent. Yeah, so thank you very much for the invitation. Very happy to be here. And yes, we're going to talk about the origin of metabolism and more precisely transamination here. So as Joris and Joseph said just before, we are indeed looking at the origin of metabolism, meaning a process that occurs something like 4 billion years ago. And in particular, we're looking at what got us from geochemistry to the earlier form of life. And we're very much looking, I'm sure you understood with Joris' presentation, we're looking at it with the organic chemistry background in these studies. I know the prebiotic field is a very, very, very diverse field. And of course, Joseph and Joris explained already why we think metabolism, the operation of metabolism is so important. And I try to add a little something to that by talking about systems theory here. So of course, life is a complex system and is composed of many complex systems. So complex systems have what we call emergent properties, meaning you have multiple individual parts interacting together to exhibit new properties. And so they are quite synergistic. And it seems like it is a very good thing. However, it's not always great to be synergistic because an issue that you can be facing is that the different parts on their own do not have any function, do not have any useful function. And you can even go back to Darwin to see this issue with the famous example of the eye, where the different components of an eye will be useful on their own. And of course, the solution here is to rationalize the apparition of this system by looking at stable intermediary stages in their apparition and looking for a continuity between these stages. For example, if you, rather than starting with an eye, you start with a few chromophore cells, which already can distinguish day and night, it's already an advantage compared to other organisms. So, and also I think it's quite important nowadays in, okay. Okay, took some time. And it's quite important in nowadays in prebiotic chemistry to look at this system approach because as we know, many of life's building block and metabolic reaction, large part of the RTCA, for example, have been reproduced non-enzymatically. And I'm not saying that it is a solved question, not at all. There's a lot of work to be done still. But we are today more interested about more complex systems, such as here, for example, the enzyme aminotransferase, which is what we are going to talk about today. When you have more than 500 amino acids linked together in a precise sequence, and that uses a coenzyme, so a small organic molecule, hydroxyl 5-phosphates. So how does this reaction work? So for the reaction catalyzed by pyridoxal 5-phosphate, you can see it basically as the sum of two half reactions. In the first one, an amino donor here, glutamate, will transfer its amino group to PLP, of course, helped by an enzyme, to get a keto acid and pyridoxamine 5-phosphate, so the amino acid variant. And then PMP can react with another keto acid, here pyruvate, to give you a new amino acid and regenerate your catalyst. And as you can see, because the reaction occurs on the coenzyme, it's where bonds are made and broken. Where people have thought for a long time that coenzyme could have preceded their protein in a proto-metabolism. And PLP is also especially interesting in this view because several cladistic and bioinformatic analysis places PLP as one of the most important coenzyme for early life. So how do we think that this process, this molecular evolution, if you will, went with PLP? So if you start very simple with an amino acid and a keto acid here, you can have them coming together, forming an adduct, an imine, and this reaction already does occur. It's only work with glycine essentially. You could call it only uncatalyzed, or we call it proton catalyzed, technically. And okay, so if you have a metal here for this proton, so the next stage, we increase a bit the complexity. You have metal catalysis, which works for a range of prebiotic amino acids. And from here to go toward enzymatic catalysis, you would like to have a PLP. So how could it have come? You can imagine having simple pyridoxal organocatalysis, PLP organocatalysis. Here, similar as before, you can imagine removing the proton and have a metal in place, so pyridoxal metal catalysis, or even more complex system with peptides or with or without a metal. And importantly, these three reactions, there have been a proof of concept that they are possible, have been known for a long time for certain of them. But the issue is that often they are not very efficient catalytically, or the studies are only looking at the half reaction or using a pyridoxal as a reagent. And for us today, we're going to advocate that the pyridoxal metal could be quite important for this reaction. And just before going to our first results, I would like also to remind you that the continuity here is a bit more important than just seeing the components are similar, because in each case, we have also have a continuity in the mechanism. For example, you can see we are always forming these imine step structures with the key step of the mechanism being the deprotonation of this position. So what was useful for the mechanism of a previous step could be useful for the next. So to compare our systems, we went back to the drawing board and we look at the reaction of glutamate as amino donor with pyruvate. So this reaction is making alanine, an amino acid. And we not use PLP in the screening, we use pyridoxal, because the issue with PLP is that you have a slow hydrolysis. We kind of mess up the slower reaction, the phosphate ester, sorry. We kind of messes up the data on this slower reaction a bit. So we use the equivalent PL, but in control experiments, they show similar reactivity. And we look at alanine formation because in this way, we are sure that we have made a complete catalytic cycle to get to this product. We compare three systems, so the organic only, meaning just pyridoxal, and this is our reference reaction. We will give it a relative rate of 1, because here we're looking at the rate because the reaction is thermodynamically controlled, so it does not really make sense to look at the yield. We compare that to the metal-only reaction and to different metal-organic reactions. So we screen 18 of the most abundant metals. And if you will, we're going to start here on the left with our reference reaction, PL alone, relative rate of 1. Straight away, we can see that when we go toward the metal-only reactions, copper and vanadium, for example, vanadium 5 here, also vanadium 3, are already much more efficient catalysts than pyridoxal, which is not really a good thing for our evolution, where you complexify your system with PL, but it's not more efficient previous reaction. We can also see that iron and aluminum are not very good catalysts here. So if we go with the PL metal co-catalysis, we have here in order of abundance. There's a few points that stick out. First here, with either copper or vanadium 3 or 5, you can see that the reaction, you can see the best acceleration that we have seen yet. And it's quite normal because if you remember, I was talking about continuity in mechanism before. So of course, what was working with copper and vanadium also works when you have PL. But on the right, we have the most interesting thing. When we have PL with our two most abundant metal in the Earth's crust, iron and aluminum, this is where we see some of the best acceleration. For example, iron was only 0.5-fold as fast as PL before when they were separate, but together we have almost 180-fold increase in rates. And importantly, it also works quite well with iron too. And the reaction works in other conditions, of course. Here we are in a very concentrated buffer at 75 degrees. It was to have a chance to observe the slower reactions. And we can do, but we don't need this buffer first or this temperature. We also look at other pH. So this time we just an ionic buffer. And we see a similar trend at 75 degrees between the reaction with either PL alone or metal alone compared to the reaction with PL and metal. And when you look this time at the buffer and temperature dependence precisely, it's quite interesting. We go back to our accepted buffer at 35 degrees. Before with aluminum, we were around at 75 degrees, around 80-fold faster. And we can see that comparatively we are much higher, 300-fold. And when we keep the same temperature and we decrease the buffer concentration from 1 to 0.25 molar, we again see quite a remarkable increase of relative rates with here 1,000-fold. So can we look at the mechanism to understand a bit better these effects? For the temperature, it's pretty simple. We can look at the reaction at different temperature and do a Arrhenius analysis. This gives us the energy of activation. And we can see that going from PL alone to metal co-catalysis, we have a decrease of almost 30 kilocalories. And as you may know, when you decrease the temperature, you're going to favor the reaction who has a lower activation energy. For the buffer effects, it's a bit more interesting. So again, standard condition, our standard mechanistic condition. This time we have only 5 mol% of catalyst at 50 degrees. That is the condition we will use for the rest of the presentation at pH 5. And what we did is that we would vary the concentration of our buffer, 250 millimolar, 500, and 750 millimolar. You can have this curve of weight compared to buffer, and we can see this very nice linear correlation in the concentration of the initial weights. Two things important to note here. Our slope is more than 0, and the slope corresponds to the buffer dependent weights. We can also see that the intercept is different than 0, and the intercept is the rate extrapolated at 0 buffer concentration, meaning the buffer independent weights, that is the rate of the background reaction by water, H plus or OH minus. So let's look a bit deeper at the mechanism to see why we see these effects. So this is the mechanism. Some of the steps are known before. They are very, very similar to the enzymatic and the metal-free reaction. And actually, because, like I said, we have two half reactions where the microscopic reversibility of each other, it's quite practical to represent the catalytic cycle as a series of identical elementary steps. So starting from pyridoxal here on the left, first step will be the formation of an aldimine. Then we have a proton transfer to go from an aldimine to a ketimine that hydrolyzes, giving you first ketoacid, the first product, and your pyridoxamine catalyst. And here we can go back in the other direction. And so you can see that our catalyst is bouncing back between these two formed, unbound form, PL and PM, which is why this mechanism is called a ping-pong mechanism. And importantly, we have observed several of these intermediates, most of them by lemma or HMS. So as I said, we have two products from this reaction. For now, we have only looked at alanine. But of course, we can also look at the other product, ketoglutarate. We have a reaction profile. Around 10 hours were done by in situ NMR. And you can see that the rate of ketoglutarate and alanine is identical. However, when we zoom a little bit on the beginning of the reaction, so after a small induction period, we can see that actually there is a slight burst of ketoglutarate formation at the beginning of the reaction. It's because at the beginning of the reaction, of course, we're not yet under steady state. So we have much higher concentration of PL that we would have during our steady state. And this is the same graph as before. We zoom a little bit in. You can see that we have around 10 micromole per minute for kg formation. But interestingly, because of course it's catalysis, we can start also the reaction on the other direction using pyridoxamine as the pre-catalyst. This time you want to look at alanine formation because it's the first product form. We can see that the rate is much higher, around 20 compared to 10 for ketoglutarate. And that tells us that the rate determining step is in the direction PL to PM, so the formation of ketoglutarate. We have a few steps to investigate here to see which is the rate determining step. We were able to rule out the alanine formation, the first two, and the deprotonation quite quickly by kinetic isotope effect. The idea here is that you would take deuterated glutamic acid at this position also, but it's far from the reaction, does not really matter here. And because the CD bonds are stronger than the CH bond, we should see an effect on the kinetics if it was a determining step. Here we have the KIE of 1, essentially. You can see here the two rates.
So sorry to interrupt. The presentation is for, oh, I see, here we are. Oh, you're back. Okay.
Okay, sorry for that. When did I disconnect?
You were going through the mechanism. You were talking about isotope effects.
Okay, that was just the slide before then. Yeah, so I'm still sharing the screen here? No. Be good.
I think so.
You want to share? Yeah. Okay. So I was here. Yeah.
Yes.
So okay. So we can rule out these first two steps. And as you can see, because the HD exchange here on the substrate, we can follow that. From glutamic acid D3 to glutamic acid D2 is much faster than the product formation, meaning that the reaction is reversible and these two steps cannot be redetermined. So we have two steps left, protonation and hydrolysis. And we went back to the buffer effect to study those. So the idea behind that being that protonation will be likely acid catalyzed and ketamine hydrolysis from the literature base catalyzed. And what we did, we performed the reaction in different buffer. We don't really care about the prebiotic possibility of this buffer here, it's just for mechanistic reasons. So we have three different buffers with different pKs. Formate, acetate, or this glycodylate, which is a arsenic acid. And each time also, and we also studied this reaction at different pHs in the same buffer. Here I just saw two of the results in the arsenic buffer. And we can see that we have always a linear correlation. And what is interesting here is that when we look at the slope, we can see that the slope, so this is a buffer dependent part of the rate, increases when we increase the buffer pK. We can also see that keeping the same buffer, arsenic here, the slope again increases when we increase the pH. It's because, of course, at a higher pH, we have a higher portion of the buffer that is deprotonated. We can also look at the intercept. And once again, when we increase the pH, we can see that the intercept increases. And it's even, as we should expect, the same at the same pH. So they will tell us that ketamine hydrolysis is likely the determining step. And to confirm that, we also were able to isolate several imines with ketoglutarate and study their hydrolysis by UAV spectroscopy to get pseudo first order weight constant. So first, if we look at the imine without any metals, this hydrolysis is very fast, around 10 minutes. But as soon as you have a metal, aluminum here, you can see the hydrolysis rate is around 265 slower. So we have a huge stabilization of our imine. And I was saying before, when we change the buffer from acetate to the cacodelates, you can see that indeed the hydrolysis is faster, which means that it is likely the redetermining step. So to conclude, we think that pyridoxal could have made an early contribution to prebiotic chemistry. And we found that metal co-catalyzed the reaction by two main effects: facilitating the deprotonation and stabilizing the imine to retard hydrolysis. It might seem weird that hydrolysis is the determining step is water. But if you think about the main fact that the metal are stabilizing the reaction is by providing a strong stabilization of these imines. Joseph said that just before, but aluminum and iron since then have also been found to improve the activity of two other important coenzymes, ADP and NADH. Another question, of course, because we don't have metal in our enzymatic reaction, is to wonder where peptide did enter the picture, if a metal was still present or not with this peptide, and that is why we are investigating now. So on that, I would like to thank you all for your kind intention, organizer for the invitation, of course, all of our group, and Joseph for having me in the lab for so long. Thank you all.
Wonderful. Thank you. Thank you both, Joris and Quentin. We'll open the floor now for questions. Now, again, you can either ask a question by raising your hand by clicking reactions and raise hand, or you can just type your question in the chat and we'll read it aloud for you. I think, so while people are formulating their questions, I have a question for Joris. So Joris, I wonder if you wouldn't mind showing the mechanism slide again, because that transformation I find very interesting.
The mechanism of the alkene hydration, right?
Yes, because I believe you invoked an epoxide intermediate.
Yeah, for the formation of the side product.
Well, so let me just put it this way. I don't dispute your results. I think it's very interesting, but as an organic chemist, I would never have predicted by just seeing your reaction conditions that one of the major products of the reaction is a redox neutral product. Because if you have iron 0 and O2, that's sort of reminiscent of like Fenton type conditions. So it's kind of surprising to me that you get this hydration as one of the main products, and that's obviously redox neutral.
Yeah.
So that's the mechanism. Oh, I see. Okay. So do you have a proposed mechanism for the hydration?
Well, I did investigate a bit the fact that I could have Fenton reaction and the formation of some hydroxy radicals in my system, and that maybe these radicals could make, a nucleophilic addition on the alkene of PEP. The thing that I did was basically I used, I'm going to show you, let me just show you 3 quick experiments that I have done. So I used the tempo as a a radical trapping reagent to see if if I have some radical species involved in the mechanism. And when you are doing that, in that case, you have only trace amount of 2-PGA while glycolate is formed. So then I was thinking, okay, maybe it's some radical species which are involved, especially in the hydration of 2-PGA, of PP forming 2-PGA. To verify that, I used two other radical trapping reagents, these are specific for hydroxy radicals, so 2-propanol and terbutanol, while tempo can trap anything. And when I'm using these radical trapping reagents specific for hydroxide, radical hydroxide, in that case, the reaction still occurs. So for me, it was hard to conclude about that. So I didn't propose any mechanism for that transformation. I can just say that we might have some radical species involved for the formation of the promoter of the reaction, or maybe directly involved in the mechanism of the formation of the hydrated product. But since I didn't manage to identify any tempo adduct, because tempo could chelate a substrate or an intermediate, and that these radical trapping reagents it seems that they are still forming the product. I didn't conclude about that, so I didn't propose any mechanism.
Fair enough. It's very interesting because I was wondering about a radical mechanism as well, like some kind of oxygen-centered radical, basically doing like it's not really a Giza addition because it's not a carbon radical, but something like that.
Yeah.
All right, thank you.
You're welcome.
Does anybody else have any questions?
Hello, I have a question. Can you hear me?
Yes, go ahead.
This is Jun Ohata from North Carolina State University. I guess I cannot turn on the video, but that's okay.
That's all right.
Thank you. I have a question for Joris. That was a wonderful talk. I also have a question about the mechanism slash like prebiotic relevance to of this reaction. I think it's really fascinating, like David said. I do believe that it's believed that the prebiotic condition is sulfur rich compared to current atmosphere, and presumably they are probably more nucleophilic to those microacceptors. Do you have some insight into that? Like hydrogen sulfide or like those, sulfur related molecules?
Yeah, so what I did, since I was thinking I have hydroxy radicals involved in the reaction, I tried to use other different of radicals. So I use sulfate radicals to see if I could promote the reaction in the same way as I was doing with the iron 0. But in that case, it did destroy all of my system. So PP is decomposed to pyruvate, and you don't have the transformation which is occurring.
I see. I guess kind of relate to that too. So O2 seems to be the major player of the reaction. Do you think other oxidants can do reaction like this too? Because there may not be much oxygen, right? Like you said. But if there's some other oxidant which may be prebiotically plausible.
Well, apart from H2O2, which could also generate some hydroxy radical in the presence of iron via the Fenton reaction.
I'm not oxidation expert of prebiotic conditions, but if there's other potential oxidants, I was wondering if there's possibility for
I
those.
tried to use nitrate instead of oxygen, and in that case, I did not manage to form the hydrated product.
I see.
And sulfate oxidant, but in that case, it didn't work neither.
I see. Well, thank you so much. It was a great talk.
Thank you for your question.
Can I just interject there about the the prebiotic relevance of oxygen? So this is something that I think for the last 40, 50 years we've kind of decided that oxygen isn't prebiotic, and because the levels of it in the atmosphere were so low.
Right.
But there's been 3 papers that Joris mentioned that have come out in the last year that have shown mechanisms by which oxygen is locally generated just from water by the flow of water against minerals. So just the mechanochemistry of water hitting a mineral as it flows splits water into oxygen and H2O2 are produced. And then just this week, there was a paper that came out showing that electrochemistry on metals that's present at the bottom of the ocean generates locally oxygen as well. So even though the global conditions may have been completely absent of oxygen or very nearly absent of oxygen, locally there may have been production of O2 that may be really very relevant. And since self-organized chemistry may have happened on the micron or submicron scale, I don't think we can rule out things like this being relevant. I'm not saying they
I
work,
agree. I
but
agree with
let's
you.
just
Yeah.
keep an open mind. That would be my kind of take on this. Yeah.
Yeah,
But
that
thanks for that
brings
great
another question to actually how much, like, I think it's difficult to measure, but how much percentage of oxygen you need for the reaction? Because if it's 0, you can't do it, but there should be threshold, right? I don't think you can easily measure, but that's something interesting to see for this reaction too.
That's a very interesting question. Yeah.
Yeah. Thank you, Joseph and Joris.
There's a question in the chat for Joris. I guess, any general comments on how the reaction is affected by salt concentration?
I didn't investigate so much the effect of the counter ion or the salt in the transformation. I think I tried just instead of sodium bicarbonate, potassium bicarbonate, and in that case you have roughly the same yield. But I didn't include any other salts in my system.
I think it's an interesting question too, actually, because if you had oxygen-centered radicals, they could oxidize chloride to form chlorine atoms, and that could do all kinds of different things. Same thing, bromide would be even more labile.
That's a good recommendation. I could have a look to that. Thank you for the question.
Are there any other questions from the audience? Well, if not, first, I just want to compliment both of you on amazing presentations. Quentin, the only reason I didn't have a question for you is because you had such a thorough physical organic explanation for the mechanism, which I was delighted to see.
Yeah, thank you.
Yeah, so it was really interesting, especially the buffer catalysis with cacodyl. So thank you both. Thank you, Joseph, for giving the introduction. And thank you, everyone, for attending. So that concludes our July session. We're going to have, let me just look at my calendar to make sure I say the correct date. Our next session will be on August 15th. We're going to have speakers talking about topics related to RNA. So thanks again, everyone. Thank you guys very much for accepting our invitation to talk. We really enjoyed it. And have a good day, everyone.
Thank you very much. It was great to be there. Bye.
Our pleasure.